Hi everyone, my name is Casey Wiesen, and today I'm going to talk about cellular function. The information in this PowerPoint is a summary of information found in Chapter 1 of your textbook. While I go over a lot of content, please refer to the textbook for more in-depth information about specific topics and place focus on the Learn Point boxes, figures, charts, and other images. There are also application to practice boxes that you may find helpful. So, Chapter 1, Cellular Function. Cells are the basic building blocks of living organisms, and the organisms can be made up of a single cell, for example, a bacteria or virus, or billions of cells like a human. And in humans, these cells work together to form tissues, organs, and organ systems. When something happens to these cells, such as an alteration, that is often the foundation for the development of disease. Since the impact of disease is evident from the cellular level up to the system level, it is important to have a strong understanding of basic cellular function and dysfunction, as it is essential to understanding pathophysiology. Individual cells are part of a network of other cells that are grouped together to serve a function and purpose, and these groupings of cells are organized into tissues. Groups of tissues then form organs, and organs are organized into systems. Cells must work together and communicate with each other in order to proliferate and survive. And when the communication or collective environment does not exist, then disease occurs. Cells can only arise from a pre-existing cell and through the process of differentiation. Cells become specialized to perform specific tasks or function. And this leads to the variation in size or shape of cells, which allows them to perform their specific tasks. Despite cells having varying functions and features, they do still share several commonalities, such as a nucleus, cytoplasm, and organelles. And there are differences among cells as well. For example, red blood cells release their nucleus during maturation to accommodate for hemoglobin and oxygen binding, and skeletal cells are multinucleate. Cells can also exchange material with their surroundings, obtain energy from organic nutrients, synthesize complex molecules, and replicate themselves. Cytoplasm, organelles, and cell membrane are the basic components of the cell. The cytoplasm makes up all the content in a cell and includes a colorless, viscous liquid containing water called cytosol, proteins, nutrients, ions, dissolved gases, and waste products. This is where the cellular work takes place. The cytoplasm contains the cellular structures called organelles that perform the work that keeps the cell alive. Table 1-1 on pages 3 and 4 discuss these organelles and describe the structure and function of each. The cytoplasm also surrounds the nucleus, which is usually the largest and most prominent cellular compartment, also known as the control center of the cell. The nucleus contains most of the cell's genetic information, or DNA, and the mitochondria contains the rest. The nucleus is surrounded by a double membrane and regulates cell growth, metabolism, and reproduction. The plasma membrane, also called the cell membrane, is the semipermeable boundary containing the cell and its components. Phospholipids are made up of glycerol and phosphate and two fatty acid tails. The intracellular and extracellular facing polar heads are hydrophilic, or water-loving, and the acid tails are hydrophobic, or water-fearing. This fatty cover protects the cell, allowing it to be permeable to some molecules but not others. Cholesterol found in the membrane maintains fluidity, and proteins that are part of the membrane act as channels to allow molecules to pass or act as receptors. And peripheral proteins attach to the phospholipids or integral proteins. Carbohydrates can be bound to proteins or lipids and function as cellular markers, so other cells can recognize each other. These markers are important for the immune system because they allow the cell to differentiate between self and foreign cells. Cells also have gates that can be opened or closed by proteins, chemical signals, or electrical charges, allowing them to be selectively permeable or allowing the passage of some substances through the membrane while preventing others to enter or exit. Having the function of selective permeability allows the cell to maintain a state of internal balance or homeostasis. Lipid-soluble substances such as steroid hormones and non-polarized particles such as oxygen can cross the membrane freely, and polarized particles such as electrolytes and large particles like glucose require specific mechanisms to cross. Hydrophobic particles are non-polarized, and polarized particles are hydrophilic. Electrolytes are charged conductors when they are dissolved in water and can pass across the cell membrane through several mechanisms such as diffusion, osmosis, active transport, endocytosis, and exocytosis. If there is a failure in these exchange mechanisms, disease can occur. Diffusion is movement of solutes from an area of higher concentration to an area of lower concentration, and is either simple or facilitated. Simple requires no energy or the use of transport proteins. These particles are small, nonpolar, and hydrophobic, such as oxygen and carbon dioxide, as they diffuse easily in and out of the cell membrane. Facilitated diffusion requires no energy and involves movement of substances from an area of higher concentration to an area of lower concentration. These particles need facilitation to move across the membrane, and this is done using transport proteins. These proteins are located throughout the membrane and are either channel or carrier proteins. Water is an example of a substance that uses a channel. While the water is a small molecule, it is also polar, and therefore has difficulty crossing the hydrophobic core. Specialized aquaporin channels move water through the membrane, and these channels are especially important in the kidneys. Ion channels move sodium, potassium, and calcium down a concentration gradient. The channels help to maintain a resting membrane potential, which is the charge across the membrane. The channels have gates to control the flow of ions across the membrane. Carrier proteins are the other type of protein used in facilitated diffusion. They are typically slower than channel proteins because they can change their shape to the target molecule. Glucose is hydrophilic and polar, and specialized transport proteins transport it into the cell. Amino acids also use carrier proteins. Water is an important solvent, and intracellular amounts must be balanced to maintain cellular health. The amount of water across various body compartments is mediated by the process of osmosis, which is the movement of water or any other solvent across the intracellular membrane from an area of low solute concentration to an area of high solute concentration. The solute concentration is the determinant of the ability to attract water, which is termed osmotic pressure. And this is what moves water across the aquaporin channels. So the higher the solute concentration, the higher the osmotic pressure, and the more water is drawn into the cell. Balance of water in the cell is critical, as too much water can cause the cell to swell and burst, which is known as lysis, and if there is too much water entering the cell membrane. And if too much water leaves the cell will shrink. Osmolality and osmolarity are units of measure of the osmotic pressure, and both measures reflect water and electrolyte balance. Osmotic pressure decreases with too much water, such as excess hydration, and increases with less water or more solutes, such as dehydration or hypernatremia. Hydrostatic pressure and colloid osmotic pressure also impact filtration and reabsorption. Hydrostatic pressure is created by water pushing the cellular membrane and forcing water out, while osmotic pressure, when it is higher, draws water in. These pressures help regulate fluid balance in the body, for example, in the kidneys. An oncotic pressure is created by plasma proteins, particularly albumin, and is similar to osmotic pressure, as higher pressure draws water in. Patients with low albumin levels, for example in liver failure, have a low oncotic pressure, and thus have the inability to draw water into the cells causing the water to stay in the interstitial space, which leads to edema. Active transport is the movement of a substance from the area of lower concentration to an area of higher concentration against a concentration gradient. See figure 4-4, see figure 1-4. The movement involves a carrier molecule, like in facilitated diffusion. However, energy is usually in the form of ATP, and this is required because of the effort necessary to go against the gradient. Primary active transport uses energy directly obtained from ATP. Three important primary transport systems include sodium-potassium pump, calcium pump, and the proton pump. In secondary active transport, the stored energy from the sodium-potassium pump is used by other molecules such as glucose or calcium to move against their concentration gradient and tag along with sodium after it has been actively moved by the sodium-potassium pump. The molecules can move in the same direction as sodium or in the opposite direction of sodium. Neurotransmitters such as serotonin and norepinephrine move using secondary active transport. Endocytosis is the process of bringing a substance into the cell that is too large to enter by other mechanisms. See figure 1-7. The cell membrane surrounds the particle and engulfs them. Phagocytosis or cell eating occurs when the process involves solid particles, and penocytosis or cell drinking takes place when this process involves a liquid. Components of the immune system, such as macrophages and neutrophils, use endocytosis to consume and destroy bacteria and other foreign material. Exocytosis is the release of material from the cell onto the cell membrane, and the material deposited onto the cell membrane will either function to replace the part of the membrane that was used during endocytosis or be released. Albumin, immunoglobulins, and hormones are examples of substances that are secreted using exocytosis. Energy is needed to fuel cellular activity, and cells can obtain this energy from macromolecules or large organic molecules. And the four types of these are carbohydrates, lipids, proteins, and nucleic acids. See figure 1-8. These macromolecules are made up of the four most common elements along with smaller amounts of other elements. The two main sources of cell energy occur from the breakdown of glucose and triglycerides. The energy production begins when the food enters the GI tract, where it is broken down into sugars, amino acids, and fatty acids. And these substances are either converted into a large molecule, such as, or an example is amino acids to proteins, and stored until needed or metabolized to make ATP. When used to make ATP, all three sources of energy must first be converted into a usable form. So, sugars, through the process of glycolysis or splitting, becomes pyruvate in the cytoplasm. If oxygen is present, then the pyruvate becomes acetyl coenzyme A. Pyruvate and acetyl coenzyme A also come from amino acids and fatty acids, and the acetyl coenzyme A from all of these forms enter the Krebs cycle, which is a high electron producing process that occurs in the mitochondria. During the Krebs cycle, these molecules go through a complex series of reactions that result in the production of large amounts of ATP. However, if no oxygen is present, the process of glycolysis will result in a less efficient pathway to create ATP, and the pyruvate will be converted to lactic acid instead of acetyl coenzyme A. The lactic dehydrogenase, or LDH, is the enzyme that catalyzes the conversion of pyruvate to lactic acid, and lactic 1 produced from this lactic acid can be used to measure the severity of a disease. Chromosomes contain the information needed when cells are replicating and information is being produced and transmitted. And chromosomes are the nucleus of the cell and contain a DNA, histone, and non-histone protein. The centromere of the chromosomes gives it its shape, and this shape allows genes to be more easily located. Nucleotides are the basic building blocks of DNA and RNA. DNA is where genetic codes are stored, and I found it interesting that if you stretch out the DNA from one cell, it would be up to 6 feet long. And if you stretch out the DNA from all the cells in the human body and place them end to end, it would be 67 million miles long. But it is so tightly coiled and wound around histone proteins in our body. And each of these units are a nucleosome, and these units fit into every cell nucleus. The chromatin consists of the nucleosomes, and 99% of the DNA is found in the nucleus, and less than 1% is found in the mitochondria. The nitrogen bases of the DNA are packed into 46 chromosomes of each cell. And when discussing DNA replication, adenine pairs only with thymine and guanine pairs only with cytosine, which are considered a base pair. And along the DNA are small parts or sections of a double helix known as genes, and multiple genes are present on a single chromosome. Genes are the physical and functional units passed on to offspring and ultimately the basis of inheritance. The transmitted genetic information forms the genotype or blueprint of a person, and the complete set of all genetic material, which includes genes, DNA, and genetic instruction, is called a genome. The detectable outward expression of this genotype is termed a phenotype. Some DNA is non-coding proteins, and the function of these non-coding proteins is unclear. But some are important and located in telomeres, which are the ends of chromosomes and serve to protect the chromosome structure from loss of genetic material or the inappropriate fusion of chromosomes during cell division. The telomeres can divide, and they become shorter with each division, which is an indicator of cellular aging, or known as the biological cell timer. Once the telomere reaches a specific length, cell division stops and growth ceases, or the cell undergoes apoptosis or programmed cell death. There are various cell disorders associated with telomere abnormalities, such as depression, which is associated with short telomeres. Other cells need to divide more often, such as bone marrow, and these cells can maintain longer telomere lengths. And cancer cells can also keep their telomeres long, allowing for longer capacity to divide. The basic requirement for life of a cell is being able to reproduce. And proliferation is the regulated process by which cells divide and reproduce. Some cells divide numerous times throughout their lifespan, and others die and are replaced by new cells. Cells do not decide to divide on their own, but rather wait for stimulus from growth factors that come from neighboring cells. And cytokines are the genetic material and protein growth factors. Genes such as proto-oncogenes within the cell direct production of growth factor receptor sites on the cell membrane and tumor suppressor genes, and some growth factors inhibit cell division and differentiation. Replication, control, and balance are important to prevent abnormal proliferation, also known as neoplasms, which can become cancer. While most mature cells can replicate and divide, some cannot, and these cells are found in nerves, muscles, and islands. It is important to note that while some cells can divide, they do have a finite number of times in which they divide before they die. Mitosis is the most common form of cell division in which the cells divide into two. These cells are somatic or non-gamete cells. Meiosis is the other type of cell division, and it is when the sperm and ova cells divide. To understand more about mitosis and meiosis and the cell cycle, I recommend you view the figure on page 14 or figure 1-14 and figure 1-13 on page 15. The cell cycle is divided into two key stages, interphase and mitosis. Interphase is the phase prior to mitosis and is the time when the cell grows and prepares for division by producing many substances such as DNA and RNA. Some cells will divide quickly, while others divide slowly. Neurons, some muscle cells, and cardiac cells have limited or no capacity to divide and remain in the G0 phase, which is the non-actively dividing phase. Also, when a cell's DNA reaches a certain telomere length and becomes senescent, the cell also loses its ability to divide and will remain in the irreversible G0 phase. During interphase, various checkpoints serve as cell quality control mechanism. After interphase, the cell enters mitosis, which occurs in 4 steps: prophase, metaphase, anaphase, and telophase. Prophase is when the chromosomes condense and the nuclear membrane disintegrates. In metaphase, the spincle fibers attach to centromeres and the chromosomes align. In anaphase, the chromosome separates and moves to opposite poles. And finally, in telophase, the chromosomes arrive at each pole and the new membranes are found. The result of mitosis is the division of one cell into 2 genetically identical and equal diploid daughter cells. Cytokinesis is cytoplasm division, and that is the final phase where the cell content separates into new cells. Meiosis is a form of cell division that occurs only in mature sperm and ova cells, and it has 2 key stages, which includes interphase and meiosis. The process of development of sperm and ova have similarities but are also very different. Sperm is continually produced from puberty, and while ova is stored in the ovaries and remain in a state of prophase for as long as 45 years. As women get older, that ova released is older and may lead to a greater risk of chromosomal abnormalities and congenital diseases. Sperm quality and quantity also change as men get older, leading to a greater risk of chromosomal abnormality, and fertilization ability decreases. Page 16 goes into great depth regarding the process of meiosis. Tissues are a group of cells with a common function, and the 4 major types of tissue cells include epithelial, connective, muscle, and neural. Groups of tissues then form organs, and a system is a grouping of organs that work together to perform similar and complementary functions. Pages 18 through 21 have tables and figures that show these different types of tissues. Epithelial tissue line the outside and all interior areas of the body, and their structure varies depending on the location and function. Connective tissue, such as adipose, cartilage, bone, blood, and lymph, are found in various parts of the body, and their most common function is to support, attach, and store. Muscle tissue is made of fibers called myocytes that consist of the contractile proteins actin and myosin. Neural tissue is composed of neural or nerve cells called neurons, whose chief function is to process and transmit information. Neurons have specialized characteristics in comparison to other cells, in that they have a cell body called a soma, an axon that conducts impulses away from the cell body, and dendrites which tend to conduct impulses toward the cell body. Cells are constantly exposed to a variety of environmental factors that cause damage, and cells try to prevent their own death from environmental factors through adaptation, which means they may modify their size, numbers, or types to manage these changes and maintain homeostasis. However, adaptation ceases once the stimulus is removed, and specific types of adaptive changes include atrophy, hypertrophy, and hyperplasia, metaplasia, and dysplasia. Atrophy occurs because of decreased work demands on the cell, which may lead to decrease in size and number of cells, and these atrophied cells utilize less oxygen and their organelles decrease in size and number. Causes include disease, endocrine hypofunction, inadequate nutrition, and ischemia. Hypertrophy occurs when cells increase in size but not number, which leads to an increase in the affected organ size. This occurs to meet increased work demands. These changes are often seen in cardiac and skeletal muscle and may be viewed as normal or abnormal changes. Hyperplasia refers to an increase in the number of cells but not the size of the cells, which leads to an increase in the affected organ or tissue size. This increase occurs only in cells that can perform mitotic division, such as epithelial cells. Hyperplasia process that is viewed as normal includes menstruation, liver regeneration, and wound healing. And hyperplasia process viewed as pathologic or abnormal includes endometrial hyperplasia, where these hyperplastic cells may become malignant. Metaplasia is the process in which one adult cell is replaced by another cell that is less mature. This is usually triggered by chronic irritation and inflammation. An example is metaplastic changes occurring in the airway. And metaplasia, just like hyperplasia, does not necessarily lead to cancerous changes, but if the stimulus is not removed, cancerous changes will often occur. And lastly, dysplasia, which is also called atypical hyperplasia, occurs when cells mutate into cells of a different size, shape, and appearance. Dysplastic changes are often implicated as precancerous and is potentially reversible by removing the trigger. Cellular death is a normal part of a cell's lifespan and may be caused by injury or a programmed cell death called apoptosis. Cell injury can occur for multiple reasons, including physical agents such as mechanical forces and extreme temperature, chemical injury such as pollution, lead, and drugs, radiation, biologic agents such as viruses, bacteria, and parasites, nutritional imbalances, hypoxia, genetics, and free radicals. In general, ATP is depleted when cells are injured, and a cascade of events leads to cell membrane dysfunction. Hypoxia is the most common cause of cell injury and often results in ischemia, which refers to inadequate blood flow to a tissue or an organ. Necrosis is another type of cell death from lack of blood supply, and necrosis tissue death from injury, disease, or lack of blood supply. Necrosis can take one of several pathways. Liquefaction necrosis occurs when caustic enzymes dissolve and liquefy necrotic cells, with the most common site of this type of necrosis in the brain. Caseous necrosis occurs when the necrotic cells disintegrate, but the cellular debris remains in the area for months or years. This has a cottage cheese type appearance and is most notably seen in pulmonary tuberculosis. Fat necrosis is when lipase enzymes break down intracellular triglycerides into free fatty acids, and these fatty acids combine with magnesium, sodium, and calcium, forming soaps. These soaps give fat necrosis an opaque, chalky appearance and occur to the breast from trauma or radiation, and also seen in acute pancreatitis. Coagulative necrosis usually results from interruption in blood flow and is often seen in the kidneys, heart, and adrenal glands. Gangrene is a type of coagulative necrosis that is a combination of impaired blood flow and bacterial invasion. This is often seen in the legs because of arteriosclerosis or in the GI tract. It can occur in three forms: dry, wet, and gas. Another mechanism of cellular injury is reactive oxygen species, or ROS, which are types of free radicals. And free radicals can cause injury, unstable agents that can cause cell death. A single unbalanced atom initiates this pathway, which can rapidly produce wide-range data. ROS are formed through the oxidation, which is a process of losing electrons, and oxygen is one of the key oxidizers. The body can generally handle a number of ROS, but excess from stress, pollution, smoking, and poor diet can lead to disease. ROS are a major influence in the development of hypertension, diabetes, and heart disease, and has also been linked to cancer, aging, and other conditions. Programmed cell death is genetically controlled and occurs at a specific point in development. Apoptosis occurs because the structure or form of the cell changes, and it is a mechanism of cell death that is not limited to certain times of development, but may result from environmental triggers. Apoptosis is important in tissue development, immune defense, and cancer prevention. Apoptosis can also result in inappropriate destruction of cells, which can lead to degenerative neurologic diseases such as Alzheimer's, ischemia, and autoimmune disorders. While apoptosis and necrosis both result in cellular death, they follow different paths. Apoptosis is when cells condense and they shrink, while necrosis occurs when cells swell and burst. These two forms of cellular death may occur simultaneously, together, or independently. So again, please refer to Chapter 1 if you have more questions or seeking further clarification regarding cellular function. Thank you.